Okay, so ladies, this is it. We're going to wrap up the concepts in genetics. So I want to finish up this PowerPoint. We've been talking about, we started with a very simple picture of genetics, right? Where we have a characteristic that is determined by one gene that has two versions. The version of a gene is called what? An allele. And the simple picture was one was dominant and the other one was recessive. And now we're starting to look at, well, genetics is not always that simple. Sometimes it is. Like the ability to taste sodium benzoate that's actually part of your lab. The sickle cell anemia is, but sometimes it's more complicated. So, for example, when we looked at dihybrid crosses, we assumed that the two genes did not affect each other. But last time we left off looking at an example where they do, where one gene does affect the other one. What's that called? Epistasis, okay? So in this example here, the gene E actually does affect the gene B. So we're going to look at now like some more examples of where it's not so straightforward genetics, okay? Where it's a little bit more complicated. Polygenics. This is where more than one gene will affect the characteristics. We've been assuming so far that a characteristic is determined by only one gene. Give me an example of a characteristic that is determined by only one gene. Give me an example. That's not a gene, that's a chromosome. Right? There's a bunch of genes on there. What would be, okay, some of the characters from your baby-face lab. Now, again, the baby-face lab, take it with a grain of salt. I don't know how accurate all those genetics are. Like, for example, the ability to taste sodium benzoate is one gene. The cystic fibrosis mutation, that's due to one gene malfunctioning, okay? But there are a lot of characteristics that they're actually the result of many genes. And how do you know if it's a result of many genes? There's actually a big clue. If you look at a characteristic and you see lots of phenotypes, and you see that it's a range of phenotypes. So I'll give you an example. Like think about weight. Okay, we have very light people, we have very heavy people, we have a broad range of phenotypes. And when you look at the range, you normally get this. Does anyone know what this is called? This pattern? It's a something shaped curve. Very good, bell-shaped curve. Okay, this is called a bell-shaped curve. And I guess because it looks like a bell. And if you see this, if you have a characteristic that exhibits this bell-shaped curve, where most of the individuals are in the middle, and as we go to the extremes, we find less and less of them. Like, think about it. How many people do you see that are like 6'10"? Not a lot, compared to how many are 5.5 or in that range. So things like height, things like weight, things like intelligence, things like skin color, these are determined by many genes, and there is a range of distribution, and it typically follows this bell-shaped pattern, but not always. We'll talk about when the bell-shaped pattern can be shifted when we get to the unit of evolution. Okay? So we can't always assume that a characteristic is only determined by one gene. Okay? Sometimes it's determined by many genes. We've also made the assumption that the environment does not affect the genetics, that they're separate. So here's a picture of a cat, basically grown. These are two cats that are supposed to look the same. They're supposed to have the same color pattern, but they're actually, because the description here is actually about rabbits, but this is actually the same thing with this cat. The difference in temperature that these cats were raised in causes them to look different. So you might think they look different because they're genetically different. It's not. It's the environment that actually produced the difference. The environment plays a role. It has an impact on your genetics. You may have the genetics that say you're going to be tall, but the environment is going to impact on that. And it may actually reduce your height if you don't get the right nutrition or if you get sick. You need to think of genetics as your potential, and then there are things that are going to happen to you that will allow you to either reach your potential or maybe limit that. So you may have the potential to be really, really smart. You may have the potential to be the smartest person in the world. But if I stuck you on an island and no one taught you anything, no one would ever know about you. Right? Think about that. No one would ever know about you. So the environment does play a role. It's very important. So, again, we're looking at genetics that is a little bit more complicated. Okay? We've made the assumption that a gene controls one characteristic, and that's it. Pleiotropy is when a gene has many effects. So, we talked about sickle cell anemia, and I specifically said it's what? Exactly what is the characteristic that is being affected? It was the shape of what? The red blood cells. But if a gene actually has many effects, we call it pleiotropy. So even though the red blood cells have an unusual shape, they have a wide range of effects. So if they have a wide range of effects, we call that pleiotropy. Okay? So it's not just one specific thing that happens. There could be a range of symptoms. That's pleiotropy. I'm going to come back to that one. I'm going to show you. So here's an example called Marfan syndrome, and it affects the fingers, the length of the arm, but also the shape of the body. Okay? I'm going to come back to pleiotropy in a second, because I want to show you something called penetrance. No, sorry. No, that was it for pleiotropy. That was it. I'm going to talk about penetrance now. Okay, that was it for pleiotropy. Penetrance. No problem. That's why you can record this and rewind. We made the assumption that if you have a dominant allele, that means what? It shows. Guess what penetrance says? It might not show. If I give you a question and you have a dominant allele, you assume it shows. Okay? What I'm talking about right now, these are like things that you should keep in the back of your mind, things that you should know about. But when you're solving problems, I'm going to give you basic problems. I'm not going to trick you and give you some obscure thing that you're like, oh my God, do I have to consider this? Do I have to consider? I just want you to know what these are. Like I want you to know what penetrance is, which I have to explain. I want you to know what pleiotropy is. I want you to know the effects of the environment. But if I give you a question on your test, it's going to be simple genetics. Okay? A simple picture. But I want you to understand what these other things are. So penetrance, we assume, and you assume on your test, that a dominant allele always expresses itself. But there are examples where it might not. So in this case, this is a condition called blue sclera. What do you think is wrong with this individual? Yeah, the eyes are... So in blue sclera, you can see that the whites of the eyes are blue. It's a dominant allele. So you would expect if someone has it, they would have blue eyes. I don't mean this part is blue. I'm talking about over here. This is blue. Okay? Yep. If you have blue sclera, you have the allele, there's only a 10% chance you might get it. Which is odd because normally if you have a dominant allele, you see it. But in this case, you might not. Okay? Expressivity is when you have a wide range of possibilities. Now, don't confuse this with pleiotropy. Pleiotropy is one gene has a wide effect. Expressivity is you have a gene, you have an allele, but it may express itself one way, or another way, or another way, or another way. So for blue sclera, you might get really dark blue in the whites of your eye. It could be a lighter blue. It could be a very faint blue. It might not necessarily be the same for everybody. So let me show you this picture. What's wrong with this picture? Okay. This is a condition called polydactyly. It's actually autosomal dominant. Don't assume that all back conditions are recessive. I'm not saying having 6 fingers is necessarily bad, but it's not what you normally expect.
Which one's the extra one? Is this the second or the third from the left?
I'm not sure.
I think it's the second or the... No, it's the third.
I'm not sure which one's the extra one. So, in expressivity, you might have... Someone who has polydactyly might have an extra finger, but they might have a deformed last finger. So when you have expressivity, it means that you're going to have a condition, but it might not always show up the same way in every individual. Which makes genetics a little bit more difficult to predict. And that's the whole point of this, is it's not always so simple. But again, on a test, you assume it is simple. Alright? Now, sex-influenced. Not to be confused with sex-linked. What does sex-linked mean? Right, it means the gene is on the X chromosome, right? And girls have how many copies? 2, and guys have 1. Sex-influenced is the gene behaves a little bit differently in males versus females. It's not on the sex chromosome. It's influenced by the gender. Let me show you something that I pulled up. This is Alessandro Moreschi, and he's going to sing Ave Maria. I want you to tell me what he sounds like. Now I'm going to see if I can get it loud enough. Hopefully you guys can hear it. Needs to go. Can you hear that?
What's wrong with him? He's sex-influenced.
How many of you know, have you music?
I know what's wrong with him. Something's wrong with his, that whole big testosterone. Something's wrong, he's not male. I don't know what's something's wrong.
Castrated?
No?
Yes. So the castrati were, I'm not I don't have a history major in music. What I understand is the castrati were a group of males that were castrated so that their voice would always remain high. They would always have that high-pitched voice. Now they stopped doing this. Apparently the guy I just showed you, Alessandro Moreschi, is the last one of his kind. Yes. I don't know. I don't know the whole story. But the thing is, when you castrate someone, I don't really need to describe that. I hope I don't. Obviously, the person's testosterone levels drop completely. So, there was a study, not a study, more like a case. There were identical twins. And one was castrated and the other one was not. Now, the one that was castrated didn't go bald. The one that was not castrated went bald. Now think about this. They're identical twins. So their genetics is identical. Right? They should be the same. Baldness is genetically determined. What happened what's going on in this case is something called sex influenced. Because the castrated twin doesn't produce testosterone, it actually changes the behavior of the gene. The gene is dominant in males because of the testosterone. But without the testosterone, it actually behaves as a recessive allele. So if you have one copy of this gene, and you have testosterone, you'll go bald. If you don't have testosterone, you don't. So, again, this is a slightly more complicated picture of genetics. Again, on your test, you just have to know what this is, but you don't have to actually calculate any of this stuff, because you're going to calculate the simple stuff, right? Okay. Here's some other stuff. Genomic imprinting. We assume that if you get the gene, it doesn't matter who you get it from. If you get Huntington's, doesn't matter if it's from your dad or your mom, it's bad. In genomic imprinting, it actually does matter. So here are two different syndromes, Prader-Willi, Angelman, different symptoms, and it comes down to who gives it to you. If it comes from your father, it's one. If it comes from your mother, it's something else. That's called genomic imprinting. In this case, it makes a difference who gives it to you. Again, on your test, you don't have to worry about calculating that stuff. Just know what it is to define it. Be aware of it. Let's skip over this. Okay.
Okay. You're posting this, right?
Yeah, for those of you who aren't paying attention, I am posting it. Claudia Palucci. Lucio.
I'm paying full attention.
What percentage of your genetics is from your mom? It's actually more.
75.
Slightly more than 50. Because there's something that your mom gave you that your daddy never gave you. Mitochondria.
Wait.
You know, well, do you remember what your mitochondria is? What's that? Everyone's mitochondria comes from their mother. Because if you remember how babies are made, the sperm cell only carries half the genetics. The egg cell has everything else. One of the organelles in a cell is called the mitochondria, has its own DNA. You'll learn why when we get to the unit of evolution. It has a very interesting story. So you have a little bit of DNA in the mitochondria. It's not a lot. It's several tens of thousands of base pairs, from what I remember. But it means that you get only that from your mom. So if there's a gene that's found in the mitochondria, it can't come from your dad. It can only come from your mom. So have you ever heard something called mitochondrial Eve? This is in reference to the last common female ancestor of all humans. And that DNA, your mitochondrial DNA, can be traced back to this person. Okay? Now, can you think of an equivalent way to trace back males? Is there something that males only get from their fathers? Or that never come from the other side? Like the mitochondria only comes from the mother side. Is there something that only comes from the father side? The Y chromosome. Exactly. So you can trace something back to Adam. No, this is not biblical Adam. This is tracing back Adam through the Y chromosome, which would be the last common ancestor for all males. We'll talk more about that when we get to the unit on evolution. So, again, there are some genes in the mitochondria, but the ones we're going to focus on are the ones in the nucleus. Maternal effect. We're almost done, girls. What's different between these two shells?
One shell is bigger and opener.
Okay, bigger and opener.
More open, same same, guys.
Spiral pattern. Are they the same?
No.
Are they the opposite?
Yes.
They spiral in the opposite direction. Turns out that the spiraling is due to a gene. No surprise there. Looks like simple genetics. Big D versus little d. Big D says spiral to the right. Little d says spiral to the left. Now, what's important is this. If you have a big D, basic Mendelian genetics would say that your shell would coil which way? To the right. However, in this case, it has nothing to do with your genotype. It has everything to do with your mother's genotype. So, if you are, let me show you an example. If your mother is big D, little d, and the father is little d, and you end up becoming little d, you would expect left. But if the mother is right, you end up being right. Now here's the interesting bit. If you're a female and you're little d, and you produce a baby that is big D, little d, maybe the father has a big D, that baby will be left. It only depends on the mother, not on the offspring's genotype. This is called maternal effect. Now, the way you explain it is really simple. Go back to genetics. What are all of these things making? No, but what does your DNA give you? What does it make? Starts with a P. Protein. So the idea is really simple here. The idea is the mother produces a protein that makes the shape of the baby's shell. So if the mother's protein says make a right spiral shell, that's what the baby's going to look like. So it has nothing to do with the baby's genotype. It's the mother's that's going to make the shell pattern. Okay? So that's maternal effect. And that's it for more complicated genetics. Okay? The rest of the PowerPoint just goes over like some disorders that we talked about, like cystic fibrosis. And there's some other ones that you can just read about, like PKU. PKU is when you cannot eliminate phenylalanine. Phenylalanine, by the way, is found in like many things, but it's found in sweetener like aspartame. Okay, so the idea is if you have a child that is has PKU and you give them phenylalanine in their diet, it will really affect their brain. Okay, they will not grow normally. Their brains will not develop normally. But if you remove the phenylalanine and you put them on a normal diet and they develop normally. So again, it's a strong reminder that you have the genetic potential to really have a negative impact on your brain if you have phenylalanine. But if you change the environment, it could change everything. Just remove the phenylalanine from the diet, the baby is fine. So as an adult, you can have phenylalanine because by that point you've already what? You've already developed. Okay? So it's a strong reminder that the environment is important too. Okay? We talked about cystic fibrosis. We talked about Tay-Sachs and Huntington's. So here's just some other examples. Color blindness. Okay? I will probably just stop there. Okay, and here are some chromosomal mutations which you can just look at. Okay, ladies? That's it for Mendelian genetics. The end.